And so of those listed, the only one that would bind hematoxylin would be the nucleolus. Everything else would bind eosin. This is an actual slide of liver tissue. This is what you would be seeing if you looked through the eyepieces of your microscope. And just take a minute to see if you can figure out, knowing what you know now about light microscopy and thin sections, knowing what you know about the properties of the stains that we use to highlight features of tissues, and see if you can identify cells and nuclei in this slide. So this is a cell, and if we basically erase the rest of the cells, we're looking at an individual liver cell. This is a good example of a liver cell. You can see a very busy cytoplasm because of all the important metabolic functions of liver. And you can also see very light areas, which are deposits of fat within the liver cell. And then just above the first cell, you can see a second cell, which is another liver cell, but this time with two nuclei. So that is a feature of liver cells. Not very many cells in the body have double nuclei. So if we look at this slide, and again, this is what we would see if we're looking through the eyepieces, we can actually see two different cell types. So there's a big, giant cell called an adipocyte, which is clear. And the reason it's clear is that the fat actually dissolved during the process of fixing and preparing the slide. So what we're seeing is a storage space where the fat used to be. Over here, you can just see pressed against the side of the cytoplasm the nucleus. So this is an example of an adipocyte. And then down at about 4 o'clock on this visual field, you can see a very narrow cell with pointy tips. And this is an example of a fibrocyte. So a fibrocyte is a different kind of connective tissue. And the name means basically a cell that produces fibers. So fibrocytes will be the basic cell type in all the connective tissues that we'll be seeing. If we look at this slide, which is the cells that we've seen before, and then one more, this slide is so, the tissue is so thin that we can actually see a second cell through the first cell. So unlike the liver cell, this is an example of a cell with two nuclei. This is an example of a very thin cell that we can just see the shadow of another cell through it. This is a special kind of tissue, so this is a very cool slide, and what we're seeing in this slide over here is a neuron. So this is the nerve cell body of a motor neuron in the spinal cord, and you can see very well a dark cytoplasm, a pale nucleus, a dark nucleolus. And then if we look at the rest of the slide again, there's another cell type there. So the active cells in nervous tissue will be the neurons, and then the smaller dark purple areas that you see around them will be the nuclei of supporting cells, or neuroglia, glial cells, which are the attendants and which serve the larger neurons. Look at this slide again and see if you can figure out which are the cells and which are the nuclei. This is an example of skeletal muscle tissue, and the cells in skeletal muscle tissue are actually collections of a number of smaller cells that fused during development. So an individual muscle cell will be multinucleated. There will be a number of nuclei in it, and those represent the nuclei of the fused cells. So a single muscle cell, like this one, can be almost as long as the muscle. So for a very large muscle, these fusions of the muscle cells can be several feet long. So these are muscle cells. So again, this is the art of seeing in histology, of knowing that the cells will be different sizes, of knowing that the cells will have different appearances, that the appearances will be determined by the functions, and being able to discriminate the features that you're looking for based on what you know about the tissues, which at this moment isn't a lot yet, but also their staining properties. And so at this point, you should be fairly good at distinguishing the cytoplasm and the nucleus from one another. Let's test that by looking at this slide of nervous tissue. The tip of the black arrow is pointing to